But give me readers. I don't really know how long now.
Hello guys, we are back with another video. Hope you have watched the previous videos and solved your queries. Please subscribe our channel and press the bell icon. Subscribe us so we can grow together. Also follow us on Facebook, Instagram and TikTok. Links are given in the description and the name is Dr. Buzz Lecture. Let's start today's topic, which is gluconeogenesis. In normal routine, when we take in food, the glucose level in the blood goes high. By the process of glycolysis, we convert glucose into pyruvate and get energy in the form of ATP. When we are fasting or doing exercise, the glucose level falls down. Then a process called gluconeogenesis helps us in making glucose again. Glucose can be made from three different molecules. One is amino acid, other is lactate, and the third one is glycerol. This process happens in liver, kidney, and small intestine. And inside the cell, it happens, which is the main question, in mitochondria, endoplasmic reticulum, and cytoplasm. Basically, there are two processes to make glucose. One is gluconeogenesis and the other one is glycogenolysis. The glycogenolysis breaks down glycogen to glucose. Glycogenolysis works in 12 to 24 hours of fasting. Today we will discuss gluconeogenesis in detail. We will discuss glycogenolysis in another video. Gluconeogenesis is a reverse of glycolysis because most of the process is same. In gluconeogenesis, we consume ATP to get useful products. So let's see which steps in the gluconeogenesis and glycolysis are same and which are not. Three steps which are not same is first, third and the last. Others are same because all other reactions are reversible. The three reactions, first, third and last, is irreversible. So there will be another way to convert them into the same molecule. So let's see at which pathway which reaction happen. First we will see how the glucose will form from amino acid, lactate and glycerol. As we know that amino acid and lactate forms pyruvate. There are total 20 types of amino acid, from which 18 amino acids form glucose except leucine and lysine. The other molecule lactate form pyruvate from the enzyme called lactate dehydrogenase. These both convert to pyruvate and then the reverse process starts from pyruvate. In glycerol, when the glucose level is low, alpha cells of pancreas release glucagon. Glucagon decreases the level of insulin. Along with glucagon, epinephrine and ACTH stimulates adipose tissue to release an enzyme called HSL, which is hormone stimulate lipase. Hormone stimulate lipase will convert triacylglycerides into fatty acid and glycerol. Fatty acid will convert into ATP and glycerol into glucose. Glycerol is also converted to acetyl-CoA by beta oxidation. Now we will see how the irreversible steps will take other pathways. Firstly, we will take on the last or the first process of gluconeogenesis from 10th to 9th step. In it, there is some process happen in mitochondria and some in cytoplasm. As we know that the last product is pyruvate, pyruvate first will be converted into oxaloacetate by the enzyme called pyruvate carboxylase. As oxaloacetate cannot go from mitochondria to cytoplasm, it converts itself into malate by the enzyme called malate dehydrogenase. Now malate can go from mitochondria to cytoplasm. After going into the cytoplasm, malate converts itself back into oxaloacetate by the same enzyme called malate dehydrogenase. Oxaloacetate will convert itself into the second product of gluconeogenesis, which is phosphoenol pyruvate by enzyme PEPCK and addition of GTP. By going through this process, there is one more difficulty that phosphoenol pyruvate can convert into glyceryl aldehyde 3-phosphate but cannot convert into DHAP. As we know that we get two pyruvate molecule, one from G3P and one from DHAP. So here glycerol take part. Glycerol will be converted into glycerol 3-phosphate where one ATP molecule will be used. Then glycerol 3-phosphate convert itself into DHAP with glycerol 3-phosphate dehydrogenase. DHAP then convert itself into fructose 1,6-bisphosphate by the enzyme called aldolase. This is 6 and 5 or 5 and 6 process of gluconeogenesis. Next, second step, which is irreversible, is fourth to third. In this process, we have to convert fructose 1,6-bisphosphate into fructose 6-phosphate. In glycolysis, fructose 6-phosphate is converted into fructose 1,6-bisphosphate by the enzyme called phosphofructokinase 1, which is also called PFK1. But in gluconeogenesis, we will use the enzyme fructose 1,6-bisphosphatase. Glucagon stimulates this enzyme, otherwise insulin inhibits. Then we will see the last process of gluconeogenesis, in which glucose 6-phosphate will be converted into glucose. In glycolysis, glucose is converted into glucose 6-phosphate by the enzyme called hexokinase. In gluconeogenesis, it will be converted back into glucose by the enzyme called glucose 6-phosphatase. This enzyme comes from endoplasmic reticulum. So this was the whole process of gluconeogenesis, in which there are 3 steps which are irreversible, which take other pathways to convert back to its same product. Now we will see more information which a medical student should know. When we take in glucose, blood sugar level and the ATP production increases. But when we consume alcohol, it impairs gluconeogenesis, which lead to no lactate, it amino acid and glycerol conversion to glucose, because of which glucose level will decrease in body. It happens due to alcohol increase ATP in the body, because of which no glucose is needed to convert it to ATP by the process called glycolysis. There will be no glucose as we have much ATP in the body, so the glucose level will decrease in the body. Thanks for watching the video. Hope you get all your points cleared. See you again in the next video. If you have any question, please ask in comment and share your thoughts. I will see you in next video. Hope we will meet again. So till then, bye bye.
¿Para qué me miras?